But bigger parts of the thalamus actually collect information from other parts of the brain, and especially also from the cortex back and distribute it again into cortex. Mm-hmm. So it is more like a switchboard rather than just a relay station. It's just not a linear relay from one end to another. It collects feedback, it distributes it again, and that I find very fascinating. Mm-hmm. Because learning is feedback, right? Mm-hmm. Learning is about, without feedback, you cannot learn. Mm-hmm. So, and somehow I think the thalamus does play a big role in that process by providing feedback in one way or another.
Hello everyone and welcome back to the Neuroscience and Beyond podcast. I'm Svilen, a PhD student in molecular and cellular neuroscience at the University Medical Center Göttingen in Germany. Along with my incredible team of graduate students, we're on a mission to bring the wonders of science, especially neuroscience, closer to you. We're excited to announce that our new podcast studio at the European Neuroscience Institute in Göttingen is slowly coming together. We look forward to hosting many more inspiring guests in this upgraded space. Our upcoming episodes were recorded during the Neurosciences conference in Göttingen. In today's episode, we're thrilled to have Anthony Holtmaat with us. Anthony is a full professor at the Department of Basic Science at the University of Geneva. His lab focuses on the structural and morphological aspects of brain plasticity. In other words, Professor Holtmaat and his team explore how sensory input and learning change the connections between cells in the brain. Join us to explore the fascinating world of memory and to learn about cutting-edge tools to study this process in the living brain. Before we begin, I'd like to mention that this podcast is a separate activity from my position at the University Medical Center in Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, the Cluster of Excellence Multiscale Bioimaging, as well as the European Neuroscience Institute in Göttingen. If you also want to support us, please subscribe to our channel and do not hesitate to drop us a comment under our videos. And now, dear listeners, please enjoy the talk with Professor Anthony Holtmaat. Professor Holtmaat, thank you for coming to our podcast.
Well, thank you for having me.
We have the Neurosciences conference going on in Göttingen, and you were one of the speakers, and we were very happy that you agreed to be our guest, and I'm looking very much forward to our discussion.
The pleasure is mine.
I would like to ask you to start by asking you, what is the topic of your research?
Okay, so I'm in neuroscience, obviously, and my focus of research is on learning and memory, roughly speaking, but mostly like focusing on the substrate of it in cortex, using animal models for this to see how experience and learning affects synaptic connections between neurons, and how that then is operating when memories are being recalled, or in more general terms, like what are the underpinnings for this type of synaptic changes. I think we will visit that topic in the coming hour. That's generally what I'm studying. We're using all kinds of imaging techniques and physiology, electrophysiology, to study these aspects. Yeah.
That's very interesting. So, how do you understand memory? You know, this is a very general term, learning. We always speak about learning and memory, and I read a lot of description of many labs, and you know, they start, we study learning and memory, and then you go and there is something completely like not intuitive. There's some proteins changes in some receptors. On the other hand, you have people who are going into a cognitive field and they like really try to understand how do you memorize these words better or whatever. It's so broad term, and when you say learning and memory, and I just want to know your opinion. How do you understand first the terms, even if you want? Mm-hmm. What is memory? What is learning? How do we define it?
This is a very good question, because I think a lot of the questions have to start, or a lot of the research has to start with defining topics and defining terms, rather than just assuming that everyone is on the same page. And I think memory is, as you say, a very, very broad term, and it comes in very different flavors. And so the things I think that you were referring to is going a bit more towards semantic memories and episodic memories, things that we are very familiar with as human beings, right? Something that we think that is what is memory is about, is that you recall something from the past. But there's, if you look really into like evolutionary into the systems, there's many different forms of memory which are still present in humans, and we all know it. We learn to walk at some point in our life, and which is based on implicit forms of memory. So these are memories that we are not conscious of, but we can do it, and our system, our nervous system, needs to be able to recall that in order to operate those functions. There's very basic imprinting and priming like memories, adaptations, very basic associative memories, which are playing out in the background in all of us at all times without us realizing it. But still, these are forms of memory, and you can study those type of memories a bit more easily in mouse models, for example, than those episodic memories. Which, yes, that's we can share those with each other because we have language, but otherwise it's very difficult to probe these type of memories in other animal systems or in other model systems than ourselves. So my research, for example, has focused a bit more on experience type of memory, which is probably more an implicit form of memory, something like has to do with sensory experiences that you want to store, that an animal wants to store in order to optimize its behavior in the future when it encounters, again, a similar type of sensory configurations.
And at what level do you study this kind of experience memory? Is it on a cognitive? Is it on a molecular? Is it on a cellular? You mentioned you're working with mice, so I assume it's more like cellular kind of work.
Yes, it's more at the cellular level. Yeah. So really we're looking at the neurons and the connections between them, so at the synapses. As soon as you get to that level, molecules become important, and you need to take into account the receptors for transmitters. You have to take into account cytoskeletal elements, all kinds of second messenger systems, which, of course, are operating at equally intricate levels as just the synaptic transmission or the action potentials that are running here and there. I think in mouse research or mouse model-based neuroscience, there's more and more behavior now, and so if you wish, you could say that becomes a little bit more at the cognitive level. We can look at which brain areas are being activated, what are the cellular, the neuronal configurations within the learning process that are becoming active, and when memories are recalled, etc., etc. But there we have to again, coming back to my earlier comment, we have to take into account and realize that we can only interrogate the system based on behavioral output, and not because of like equal level of sharing of information that we can do with animals. So
exactly. So maybe there is something else even that we don't know, but since these animals cannot communicate it, we just have we've been left with what is out there in terms of behavior.
And there is, I think, lays the big caveat or trick the problem in a way is that we need to somehow interpret, and in doing that we often anthropomorphize the behaviors. We think we completely understand what for example, what the goal is of an animal when it's trying to do something. For example, if you let it operate in a head fixed go no go task, we assume that all it wants is to get as much water, for example, as it can get. But we often don't take into account that there may be some level of saturation or like attendance problems, or other. Maybe it's just ultimately wants to go out of that situation as soon as it can, and it has figured out that by doing this and this and this, it can get out of it quite soon. So it's really, it becomes a bit of messy operation, I think, if we go there. But it's interesting. It's interesting enough because we could still, infer and try to deduce things from what we think is going on at the behavioral level. Or
I think it's a very important comment that you made about the I cannot pronounce it. Humanizing of the behavior, right? So we think that, because we behave this, they should also behave this. But this is maybe not the case. However, we still draw some valuable conclusions that help us learn about our behavior. Then. Can we agree that we can study like learning and memory in terms of, let's say on the macro scale in humans, but we cannot do it on a molecular scale. Therefore, we need the animals.
Yeah, I think that's a very good point. We, I don't know. Well, I mean, that also assumes a bit that all we want to know is things about ourselves. And I think this is also not necessarily my goal. I of course, I would love to understand all what's going on in my brain and other people's brains and how human beings learn and memorize. But it's equally interesting to look at it from an animal's perspective. And again, coming back to what one would call an animal's Umwelt, right? What is important for this particular individual and this animal or this species to operate in the world? And I think this becomes even more relevant while we are dealing with climate crisis and with we are losing species on a daily basis. We really look at the world in a very, very anthropomorphic way and just reason from ourselves by understanding animals. This is a German word, I guess, Umwelt. There's, by lack of a good English term, I think this is really often used, I think, in science, this word. We may also maybe improve on that in that realm. So anyway, this is just a little sidestep. I think this is not just only to understand the human mind.
Yes, but it's important to be said, I think.
Yeah, absolutely, yeah.
And also, of course, we can study the fundamental principles of nature, right? Not only animals, but how systems operates in this regard. And it's just incredible that we have the tools nowadays to do that. And speaking of tools, you're using some very, very interesting techniques like the two-photon microscopy and the calcium sensors in the brain that kind of help us track brain activity on a large scale or on a cellular scale, and other tools that you probably will share with us. But how do you employ them to study, for example, the things that you're studying, memory?
I think this is... Well, it's one of the tricky things as well in science or in neuroscience. It's very much technique driven. I think there's lots of questions and people have all kinds of ideas on what one would like to study and how one would like to study it. But very often we simply lack the tools, right, to go in there and to record cell activity, neuronal activity at large scales. We just don't have the tools to know exactly what's going on at synaptic level. And it's getting better. And often, a new technique comes up and suddenly you see all these questions are starting to pop up as well. And almost looks like these questions are new, but they're not. They're just, they suddenly can be addressed. And so, we are doing the same. We apply new techniques when they come about. We try to help a little bit in the development, but my lab personally is not very pioneering in that respect. I was a bit at the forefront, I think, when two-photon microscopy started to really take off and started to be applied in vivo. So, I was fortunate to experience that firsthand. And it really showed me the power of really seeing things, live happening. That said, there is always this uncertainty principle when you look at the brain. Is this really, what is going on when you don't look at it? Is it generalizable? Can you apply it to all systems? Let's say in cortex. So yes, these underlying doubts always remain. Same with measurement of activity with calcium imaging. Calcium is an ion that, a flux of ions that is important during activity, but it's not showing everything. And it's usually not well showing what's going on under the hood, as one says, right? There is a lot of computation perhaps happening in a neuron or in dendrites that remain invisible. I was very excited when I saw that the tools coming up with which we can image the synaptic activity, because that really gives us a first possibility or glimpse of how activity of a neuron is related to the activity of synapses, which we so far could only do with electrophysiological measurements, but we couldn't really, not really do it at the synaptic level. And we could only infer that particular synapses or a number of synapses was active. So yes, lots of exciting technical developments, both at the physiological level and at the imaging level. And collectively, I like to term these optophysiology. I think that sort of that groups everything together, and all the tools like optogenetics, calcium imaging, physiological recordings, I think altogether would be optophysiology, which is the new sort of level of techniques that we have right now.
We are moving from the electrophysiology, which was partially developed here from a person who is Göttingen, Erwin Neher, who actually is one of the people who initiated the building of this institute. We are currently at the European Neuroscience Institute in Göttingen, and this was, I think, an initiative that started more than 20 years ago, and now we're still here, and people still learn neuroscience, and it's great. But back to the topic, the optophysiology, if you want to say it. Two-photon microscopy. Can you elaborate on this technique and on the calcium imaging? How does this work? And why do you use two-photon microscopy to image, for example, in vivo mice in vivo?
Yeah, so two-photon microscopy is basically a sophisticated form of microscopy, which is not in essence not very different from other types of microscopy, in the sense that it relies on fluorescent proteins or fluorescent dyes that you can inject in the brain, and fluorescent proteins you can express with genetic tools, with viruses or with transgenic mice, for example. And these fluorescent molecules can be readily excited by light of particular wavelengths. The problem with regular, let's say, confocal microscopy is that you need quite some light to penetrate brain tissue, to go deeper than just a few microns, for example. And so that poses a problem if you want to do it in living material, and especially if you want to do it in a living brain through a cranial window, for example. And the other problem, and because you need to apply quite some light, you are causing toxicity, and you will have to filter out all kinds of background light that is coming back. So two-photon microscopy solves a lot of those issues in one go, because only a very small focal plane will have sufficient a number of photons basically to excite the fluorophores. And so you get rid in a way of the background problem, and the higher wavelengths that you use typically for two-photon microscopy is a bit more forgiving to the brain. It's less toxic, and maybe the fluorophores are less easily quenched or producing non-desirable phototoxic effects. That said, it's not it's not the golden bullet. It's there's still you can still make lots of mistakes with it and do things that you're not intending to do. So in terms of pathology or toxicity.
Yeah, for people who are not familiar with this concepts like confocal, etc., the thing that we were discussing is that in principle you have a focal plane that is illuminated, and when you use the so-called confocal microscope, you illuminate also the above your focal plane and below, but because of the technical properties of this microscope, you will kind of get rid of this out of focus light. However, you will not record it, but the cells will be illuminated, meaning that
there will be light going through this tissue, obviously.
Yes. But in two-photon, you have this only in one plane because of the nature again of the tool. That these two-photon have to be at exactly specific time to meet each other at this point, and the sufficient energy should be there to excite the dyes, right? So therefore, you have this kind of excitation in a certain plane, without kind of illuminating and damaging additionally cells that are above or below. And of course, the other huge advantage is that you can penetrate tissues quite deeply, right?
Yes, because the wavelengths are usually, let's say, typically twice as high as what you would do with single photon. And that usually is a bit, that's more in the red spectrum, and that allows further penetration, deeper penetration. So it's not still, not crazy, but you can get a bit further than otherwise.
It's enough to address certain questions.
Yeah. The disadvantage is that you need expensive lasers to create this this dense enough packages of photons to get that excitation going.
Yeah, these instruments can go up to, I guess, million.
When you're talking about very, very sophisticated, but you 100,000, 200,000. It's not unheard of too.
It's expensive, and it's also hard to operate. You have always this misalignment of the lasers, right? So from time to time, it's
getting better. Yes, I think when I was in Cold Spring Harbor with Carl Svoboda, we were still using lasers that needed some manual tweaking, and that was part of the morning would go into trying to get things stable. But over time and very rapidly, when the industry started seeing a market, I think they started to develop more off-the-shelf or like turnkey equipment. So nowadays it's a vacuum chamber that you don't open and you just switch on the key and it works.
So I was there, by the way, recently in a Cold Spring Harbor, taking part in a microscopy course. Shout out to the quantitative imaging course, from acquisition to analysis. Very good one. Where is his lab located? Is he still active there?
No. He has since then moved to Geneva Research Campus at some point, and now he is at the Allen.
Okay. I was surprised by the campus at Cold Spring Harbor. It didn't look, that impressive. There were like these nice houses where, is this a lab now? Is somebody living here? But apparently,
some Alzheimer's can just be fast cars.
Researchers,
I always loved that campus. I thought it was fantastic. It really had such a good time there.
It's nice and quiet for people who haven't been there. It's really, really great place to do science and not be distracted at all.
And it has so many perks. It's close to the water. It's beautifully located. It's close to New York. I mean, for me, maybe I'm romanticizing it a bit, but I really, I had such a good time there, and I can recommend it to anyone to at least visit,
go for
take a course or go to a meeting.
It was fantastic indeed. So this is one of the tools, so the microscopy. And then you have the genetically encoded calcium indicators, if you call it. The GCAMs are part of this group. How do you combine these two for study?
Sorry, which two?
The two-photon and the calcium.
So of course now when we are measuring structure, when we just have, let's say, the green fluorescent protein, jellyfish protein that you express in neurons, and it's just it's just expressed in neurons, and it's quite stable. So you don't have to worry so much about the temporal noise or artifacts. Now, when you are doing something dynamic, it becomes all a bit more trickier at the acquisition side, right? You need to acquire fast, so you need to make sure that you have good detectors that can, detect photons rapidly and are sensitive. You would have to, also scan rapidly. So it becomes a bit more, quite a bit more tricky to do it right. And on top of that, there's, also the proteins are still developing. The proteins are very notoriously slow. So you maybe miss action potentials. You overinterpret the signals. The signals have long tails, so it's difficult to really, peel off the actual activity that is underlying it, right?
When you're saying that the proteins are slow, you mean that from the excitation to the detection, the process is longer than a one action potential?
Well, it has to do with different aspects. It's also in part the excitation, but also the just the sensitivity to the calcium, right? That you plays into it. It's an artificial molecule, if you wish. Right? Of course, it's built on components that the cell has, calcium sensors that are being re-engineered to then create fluorescence by attaching GFP to it. But these conformational changes are slow, and sometimes they remain in that position for longer than you wish, and so there's issues at all kinds of levels. But there is, like at Geneva Research Campus, they have done an amazing job in generating better and better versions. So now I think nowadays we really are at a high level of sensitivity and also of temporal resolution is very high now. And I think they're rivaling by far now, or they're outperforming some synthetic dyes for sure. So and that's of course every time there's a new protein coming up, you have to go through calibration rounds. You have to retune your settings to this and see if everything is fit to really work with that particular probe. So it's not just like click and go. It's
not a straightforward process at all. Yeah. So we have now the two-photon microscope that helps you to excite this kind of fluorescence protein, and then some detectors or camera, depending on the microscope setup, that detect it. We have the proteins and the calcium indicators that will eventually show you some, it's not direct activity, but it's a proxy of activity, right? And then you have the mouse or a rat. I don't know what exactly you're using. You mentioned actually. Mice. Yeah, exactly. And how do we look into the brain? There are these cranial windows. What are these things?
Yeah, so that was also quite some... it sounds so simple nowadays, I think, but it was not always straightforward just to, take a piece of the skull out. That's basically what it is. You very carefully remove a little bit of skull and you replace it with a piece of glass. But of course, this is easier said than done. You have to make sure all your surgical methods are correct. To do it chronically, you need to find ways in which that, like temporarily invasive surgery is tolerated and doesn't lead to degeneration. So it's there's a bit of tinkering that is still going on, and these techniques, these windows, are still being improved with very sophisticated ways of like crystallized skull, like putting half of the skull, replacing it with something transparent. So yeah, it's super exciting that this is all possible, and yeah, in a way, the name says it. You create a little window in the cranium, and the mouse dura, the dura mater, which is like overlaying the brain, is translucent or transparent enough to allow the light to go through, which is it's really fortunate that is possible in a mouse brain. In a rat brain, it becomes very, very difficult. And usually the bigger the species, the thicker the dura is, and then there is a need to remove that dura, which can cause like long-term issues. So you really typically try to leave everything intact, only take the skull off. And in a way, that's also what happens in human beings. When they need brain surgery, there's usually a piece of skull removed. So yeah, it is tolerated, but you have to be careful.
I guess some inflammation is happening when
you could have that if you don't do your surgeries carefully. Then that could happen.
The dura mater is basically one protective layer.
It's a very important protective layer that you prefer to keep intact. But which is not always possible.
Yeah, in humans it's very, very thick.
Very thick. It's almost thick. You cannot even go through it with you.
We had this in anatomy, and it was fascinating to actually hold a human
it's like
brain,
a piece of plastic almost.
And the dura, and all memories, all emotions, all everything in like, I don't know, 2.5, 3 kilo or whatever is this mass of
Isn't
cells.
it fascinating? Yeah.
It is indeed incredible. And the question now that I'd like to ask you is, can you image mice for longer periods of time using these 2-photon and cranial windows? Or do you do it like more acute in 2-3 hours, then you stop? What is the experimental part?
No, I think the real the advances that we made back in the time, we're talking about 2002-ish, 3, 4, those times, was really to get this to work for long term. An acute prep is you can also take the dura off and do something acute. Works really well. But you get into trouble when you want to then extend it. And if you want to repeatedly revisit a particular neuronal structure, you would really need to have a stable prep, right? That doesn't really induce a lot of changes by itself. Now, again, there's an uncertainty problem. You don't know exactly, would these same changes happen if you didn't do anything? But of course, we have to look in one way or another. But we have done lots of controls and lots of experiments, and things have been verified with different methods. So yes, I think with different types of cranial windows, you can see similar structures over time, over weeks, months, and some people have even taken that 2 years, year, I must say, for a mouse, for example, and look at synaptic structures over time.
So speaking of synaptic structures, what are synapses? These are very interesting structures in the brain, right? Can we assume that they are like the biological unit of memory? Can we
that's a very now we're getting deeper. I think I wouldn't go as far as that these are the elements, or they are very important elements for memory and learning. I think synapses are present in almost all living organisms with nervous systems. Some people even claim sponges might have something like a synapse structure, and some plants might even have synapse-like operating communication devices. So, and they get more complex the more complex or the bigger the nervous systems get. So, I think learning and synapses go hand in hand. I think there's something important about them. But that said, I think there's other elements in neurons that we shouldn't forget, and other like non-neuronal elements perhaps that are also important. I don't want to dismiss any of those. I think we should keep an open mind. Just simply, the excitability of neurons that can be regulated intrinsically, those could also harbor certain memory mechanisms or learning mechanisms, and also molecular expression profiles. Maybe within the same set of neurons with different set of proteins, you operate differently. Mm-hmm. So I think we can see it. We can see this like learning taking place at different levels of the circuits. But nonetheless, synapses do play an important role, I think, and that this is universally accepted, but not by everyone, but I think by majority of people. Yeah.
So synapses are these places where the two neurons communicate, right, with each other. Or there was a recent paper in Science, and they did this connectomics. I mean, it was a week ago.
Connectomics, yeah.
Where they discovered also in human, if this was a human tissue, neurons having connections with themselves, which we study in our labs. It's called autoptic cultures, right? Yeah. But I don't know, is it known in animals? Probably.
Well, I don't know. I think we haven't really. There's autopsies in I think, even with simple tracing or techniques and immunocytochemistry and neuronal filling, and even electrophysiological recordings, you can see autopsies. Mm-hmm. Now, how common are they? Usually they're considered not to be very common. They happen, they're there. What is their role is also not very clear. Mm-hmm. Now, it turns out in this study, indeed, that in human, there's a lot of them. Mm-hmm. So does this have a specific function? Is this something that evolved that way? Or is this a byproduct of neurons trying to connect with as much as other neurons as they can, and sometimes that happens to be themselves. Mm-hmm. Now, an autopsy is an interesting unit, if you like, from the perspective that of Hebbian plasticity, because Hebbian plasticity would state that what fires together wires together, and synapsing with yourself would automatically mean that you fire together, right? Mm-hmm. But there's probably something that prevents these things from happening a lot. So that's, I don't know. Maybe some people have studied this in more detail. I have not really studied this, but that would be my guess, that there are some active mechanisms that prevent that from happening massively. Mm-hmm.
Yeah, definitely there should be some computational aspect about how this feedback and but it's, as you said, it's a different topic. But one of your topics is the study of protein lifetimes. I don't know if you still work on this.
I have done not a whole lot on it. Yeah. I think back in Cold Spring Harbor there was. Noah Gray, who is now editor at Nature, he was, together with Robbie Weimer, doing some studies which I found very interesting, and I followed it at the time. And then I picked up some of the loose ends that were left after that study was published. But unfortunately, that was with overexpression systems. So we had to express a synaptic protein, PSD95, which is a scaffold protein in the synapse, tagged with GFP, and then you can follow basically where that GFP goes. You can also have photoactivatable forms of it, and you can turn the fluorescence on and see how long it takes for it to disappear. Mm-hmm. Since it's overexpression, it's not very clear if you are really mimicking or emulating the real endogenous situation. So nowadays, I think there's much better techniques. And recently, we collaborated with Seth Grant, who generated a mouse line in which a tag, halo tag, was knocked into the PC95. So then you're embarking on the endogenous expression mechanisms to get that tag in there, and then you have ligands that you can inject, fluorescent ligands that are inert, so you can then follow, basically, once a synapse is fluorescent, you can see how long it takes for that fluorescence to disappear. Now, there's all kinds of calibration and controls that you need to do, and see if you saturate the tag with the ligand, if you don't change the dynamics by illuminating it, quenching. There's all kinds of things which is still all a bit like we're still not completely out of the woods there, I think, but it is already starting to show interesting things that sometimes these proteins stick around for a long time. So maybe surprisingly long. But for some, it also shows that very quickly it turns over, which is also fascinating, because that means that, the structures are maintained despite the fact that the proteins are disappearing and being renewed and just either moving to other locations or being degraded. And I think that also fascinates many people. It's like how, in general, how do you keep things stable with so much plasticity, if you wish, with so much change, and this drive to entropy that is constantly happening, and the system is fighting to this entropy and trying to keep things stable. That by itself is a level of plasticity, I would say, right?
It's very interesting that you have indeed this kind of memories, but incredible turnover of proteins. Yeah. However, you maintain the same memories, or if we assume that they're in some synaptic circuitry, right? Although you have this protein turnover, they kind of are there and maintained in some...
the protein turnover and probably with some... stochastic events will lead to disappearance and appearance of synapses. We've even seen that there is quite a bit of baseline activity going on in terms of new synapses that are being formed, synapses that are being removed. It's not always clear that this is a very active process. Maybe at some point things are just simply with the protein levels they have, or the protein distributions, things can just simply not be maintained because the hardware is not there, and so you lose synapses. But despite that, apparently, the functionality of the circuit remains rather stable. This is I think also from a theoretical and computational standpoint, interesting to see how that is possible, and it must be that. A lot of these functions and memories, if you wish, are distributed. They are not just dependent on, a few synapses. They are dependent on some sort of coherent activity that can arise, and which can arise from in many different ways. And right.
Indeed, and as you mentioned, we are talking about here the synapse and neurons, but we have also other cells, right? The glia, which definitely contribute to all of these processes. And I personally study the extracellular matrix, so this is also another aspect on top of this kind of structure that helps stabilize synapse or help keep them kind of together. Let's put it that way. So it's incredibly complex to study, and a synapse that is, what is it, like, I don't know, some nanometers, a nanometer range, right? And you have tens of thousands of proteins, right? And they interact with each other, and it's incredibly complex.
Yeah, it is interesting when you do these type of experiments, and you can fluorescently label, and hopefully now in the future we can with CRISPR Cas9 we can actually tag endogenous proteins with fluorescent, or with tags that can then be fluorescently labeled. And hopefully then we can really get a good view of the dynamics, right? But the first things that we've done already 10, 15 years ago or 20 years ago is showing that there is a lot of dynamics, a lot of change going on constantly. Also, if you look at cytoskeleton, for example, it's constantly being renewed and broken down. So, this is a lot of the homeostatic forces that are or homeostatic processes that are happening. I think we shouldn't really dismiss those as just simple, household processes. There's actual active plasticity going on, and this goes again hand in hand with forms as Hebbian plasticity. You cannot have one without the other. I think there must be always some level of equilibrium or balance that the system arrives at. And otherwise, you would just make more and more synapses, right? Until the neuron is completely saturated with synapses. Apparently that doesn't happen. There must be some mechanism that prevents that. There must be some mechanism that prevents an excessive amount of autopsies, et cetera, et cetera. So, that interaction is also very, very fascinating. And the extracellular matrix, when you mention it, it also fascinates me now because some recent findings that, if you look at seemingly similar synapses that are quite close to one another, for example, they can have different protein contents. And how is that regulated, right? How much are the pre- and postsynaptic elements talking to one another and providing signals through the extracellular matrix probably as well to recruit those proteins and to create some level of specificity. That's a very fascinating aspect as well, yeah.
Yeah, the ECM is, or extracellular matrix is very unexplored field, and
it's difficult to study.
It's difficult to study. Yes, I can confirm that. The organization and the nature of the molecules is it's a little bit more challenging to approach from this kind of tagging perspective, for example, which kind of hinders you to use sophisticated imaging approaches and look at the dynamics of these molecules. But we are working towards
good
direction.
Glad to hear.
So I wanted to ask you something about the sensory and input and memory. But before that, just another question that came to my mind. We have cells in the cerebellum, for example, that have up to 100,000 neurons on them, right? And
spines, you mean?
Spines, synapses. And then we have some cells in other domains of the brain cortex and hippocampus that they have less, let's say up to 10,000. Why is this? Is there any reason why this have?
It's always difficult to ask for reasons. I think things evolve as they evolve. Maybe there's not much reason. It's just that things don't get punished, or they provided some benefit, which you could translate into a reason. So I wouldn't know. I wouldn't know why this is the case. I think we're getting a bit closer also for cerebellum in trying to, find the computational elements. Right? What is the computation that is being done by the circuits? Right? And for cortex, we're maybe still a bit further off, but that might provide some clues as to why you have so many of those contacts. This also might depend on how these synapses integrate with one another. Maybe in some dendrites or some cells, the integration is linear, and maybe you can have, quite many of them, and you just basically linearly summate, the incoming inputs. Other cells, in my opinion, like pyramidal cells in the cortex and also in the hippocampus, have these local dendritic computations or integration that is dependent on clustering of certain synapses, some regenerative events that happen, voltage-dependent conductances that occur. And maybe you just need a few of those in order to evoke enough a large enough conductance that reaches the soma to reach the action potential. The threshold. So I mean, that could be a reason, right? That there's differences in intrinsic differences in excitability that allow more or less synapses. But who knows? There might also be some very, requirements at the computational level that we just don't know yet.
Something completely different that we haven't thought about it. But sure. Now back to your research, the sensory input and learning. So what kind of sensory modalities are you studying? I know that people use in mice, especially the whiskers and odors. I don't know what is exactly.
You did your homework. Yes, we study the whisker, or let's say the somatosensory system as a modality. And in mice, then you very quickly arrive at the whiskers because it's such an important modality for them, or at least what we think. And why not? Because it's so prominent and it spans so much space in the cortex relative to other modalities. So they use that system quite intensely to navigate their surroundings. But surprisingly, we know surprisingly little of it, really. It's an active sensation, so the animals, to use that properly, the animals have to move their whiskers, which makes it difficult to study it. And so that's immediately a disclaimer for what we do, because we often, let's say I call it yanking often, and we pull or we tickle those whiskers, but of course, not in natural ways. So the interesting aspect of it, or the benefit of the system, is that we have a good anatomical understanding of how these whiskers interact with the cortex, and how the signals are processed. Like every whisker has a relatively large area devoted in the cortex to its processing of its sensory signals. And so we have the idea that we know where to go, where to look, and that's the important part when we tick on a whisker, when we tickle a whisker. I think it's, for me, on the one hand, I'm interested in knowing how that system works, how it is functionally operating. On the other hand, I also just see it as a model system for maybe more to get to a better understanding of the microcircuits that are operational in cortex, which to some extent are perhaps let's say, I don't know if that's the right word, but universal, are like you can probably generalize some of these circuit properties that you find in the somatosensory cortex, in this case the whisker barrel cortex, it's called, to, let's say, visual cortex or auditory cortex, or maybe even non-sensory cortices. So I think for me at the moment, I think that's the way I look at it. It's a nice model system that we understand anatomically quite well, and functionally. In the context of behavior, not so much. But,
So to put things in perspective, let's imagine a setup, and you have a mouse with a cranial window with a two-photon, and then you can stimulate the whisker, and then because you know exactly where this whisker or the sensory cell relays information to exact location in the brain, in the somatosensory cortex, so we can observe the events there and study them. Yeah, this is correct, right?
Exactly. Yeah.
Yes. And then you can study also circuitry that are around this one and see the connectivity eventually.
Yeah. So if you let's say we pretend we know what we're doing at the sensory level, so we tickle a whisker. We do this very briefly, plop, and then there is a signal going into the cortex. Now we can reproducibly create that type of activity. We can now go in with optogenetics or with other means, or we can do recordings and see what exactly is going on at the synaptic level. What if we block the activity of one particular afferent input? What if we overexcite particular inputs? Does this change the way this sensory signal is being processed? And by that, we hope to get a little bit more information on what are the governing principles of this sensory processing.
Fascinating.
Yeah.
The toolbox is growing and growing and growing.
Absolutely.
And you can address even more questions and more questions. And I think there is a lot to do in terms of understanding the network's dynamic, right? We have kind of this discrepancy. We have this molecular aspect, the cognitive aspect, but in between it's still not there yet. We still kind of develop tools, and I think this is one of the methods that can help us understand the brain in more like the circuitry level, which is crucial to understand the higher cognitive processes, right?
Absolutely, yeah.
So a lot of the sensory information that we get from the world, the visual, the auditory, smell, haptic sensations, this information goes from our sensory cells through the thalamus, which is a very interesting structure in the brain, and then goes to the cortex, probably one of the areas that maybe long-term memory are stored and the planning is going on. Higher cognitive functions are in this kind of field. What is so important about the thalamus? Can you tell us?
It is an enigmatic structure by itself. I think, and maybe as much as the cortex. I think, they go hand in hand, right? All vertebrates have both. And so it is something that also grew in complexity over time, let's say, during evolution. And so, there is something about it, which I think for a long time it's been seen as just a sensory relay system. And of course, that I think by now is clear that we have to depart from that view. It is not just simply a relay station for incoming sensory signals. It does that, but bigger parts of the thalamus actually collect information from other parts of the brain, and especially also from the cortex back and distribute it again into cortex. So it is more like a switchboard rather than just a relay station. It's just not a linear relay from one end to another. It collects feedback, it distributes it again, and that I find very fascinating. Because learning is feedback, right? Learning is about, without feedback, you cannot learn. So, and somehow I think the thalamus does play a big role in that process by providing feedback in one way or another. And so this is something that we were trying to understand a bit better. And so yes, but why is it there? Why was that? Did that structure evolve as it is? We don't know, but it's really, I think, goes hand in hand with the cortex. I think that's it's as much as I could say about it, I think.
I allow me to correct myself. I said all sensory information, but the auditory,
the auditory does, but the olfactory.
Olfactory. Olfaction, it has a direct connection to the piriform cortex, right? So they also have.
Interestingly, the olfactory chemical senses, I think, that already were there in very early invertebrates and worms. They did it without thalamus. There's all kinds of ideas about what makes the other senses so different. What did I read? I read once that some people were arguing that maybe the invariants that you have for most senses do not apply to olfactory system. Like we all have abstract ideas of what a glass is, right? If I put it upside down, I still can know that it's a glass, or if it becomes miniature, I still know it's a glass. And I think many mammals, for example, can have that capacity. And it's the same with hearing, and the same with haptic information or some of the sensory information, and maybe not so much for olfactory chemical information. But who knows? I mean, it becomes all a bit speculative, and it's an interesting hypothesis. But,
exactly.
Yeah.
Why do people consider the thalamus as the gate to consciousness, for example? Do you know that? I mean, I guess it has to do something with sleep. And
yeah. Well, it's a very important in sleep, and it definitely, there's parts of the thalamus that regulate it and that can induce it. So yeah, again, because it has those, it collects information from cortex and sends information back. So also things that we've seen, not so much in context of sleep, but some parts of thalamus and the higher order thalamus in somatosensory system, we find regulates excitability of cells. And so it might have a certain switch function. If it's on, these cells can operate and they can integrate other information. If it's off, maybe these cells are going into sort of like sleep mode, if you wish, and become less excitable, and other signals have less influence over them and drive them less. That's a very simple way to put it, but that could be a way thalamus or thalamocortical projections function.
It's
Not the primary relay neurons, but the ones that actually feed back information.
And it's a kind of a station where during sleep, for example, it shuts down the connections to the cortex. Therefore, we don't leave our dreams, right? And it's a very interesting structure to think about. If we can go back to memory, I read a paper, I don't know what time ago, but it was about introducing false memories in a mouse model. Right. I don't know if you're aware of this study.
I think there's by now quite some studies in which memories are being manipulated in mice.
How does this work? Can we do this in humans already?
Yeah, it depends how you look at it. I think, for sure. We do it in humans on a daily basis, but we don't do it with tools. Brainwashing is a very ancient term and has been done for forever. And in a way, that's what it is. You feed information and let it compete with other types of information. If you do it long enough, you install something that's not there, and or you modulate it in such a way that you have, let's say, changed the memory. And I think, we are so, our memory systems are so valuable. It's not like we always think that, okay, we have the absolute truth when I memorize something. It's really dependent on so many other information or processes that are going on, the internal states, and our cortex and our brain fill in a lot of information that we think is there, but it's actually have never been there. And we all these visual tricks that we know from video games where some ape is, going through a basketball court, but when people are, not paying attention to that, they just simply don't see it, and vice versa. So I think, false memories are will always bother us and will always be present. Now, how you control it in an active way, and I think that's what you're referring to, right? That you actually have some tools that you do something at a molecular level, or with, injecting activity in circuits to create a memory out of the blue. I don't think, I'm not so sure. Probably things have been done in humans, but I don't know how reproducible these things are. In mice, yes, because we can go back to those very basic memories that I talked about earlier, like implicit memories, associative memories, especially associative memories can be tinkered with, right? If you embark on or recapitulate place cell activity, you can actually create a memory or modulate a memory that's false. And this since the optogenetics have arrived, I think these experiments have been done by many people. And it's interesting. It always speaks to the mind or to the imagination, right? That this is actually possible. It also, is a sort of an interesting and comfortable confirmation for us that we're looking at the right thing, that we look at neurons, and that this is actually where, and the synapses, this is where memories are being, let's say, stored if you wish, and learning is happening. So
yeah, this is the place cells that they fire when you're at a certain place, right?
Yeah.
And you can trace back and
if you then know the patterns that are being activated and use molecular tricks to replay that activity, or prevent that activity from being played back when a particular memory has to be recalled, and then mice fail to do that. So these are very fundamental, important studies that show that also that place cells in the hippocampus, for example, is important for this type of associative memory formation.
The guys that discovered this, they got the Nobel Prize, I believe. They studied this kind of, there was a study already in mice.
Place cells,
and then there was this interesting study about the taxi drivers in London, that their hippocampus was enlarged.
That's also that. Yes, true. Yeah.
Do you use optogenetics in your work?
We do, yeah. Actually, I find the optogenetics have an incredible reach, and they've penetrated all corners of neuroscience. And it's a very powerful tool. Sometimes we overuse it maybe a little bit, but I find it interesting to use it just to simply study synaptic circuits. We can still, we can really use these tools now to play activity, right? In a way that we couldn't before, when you had just simply electricity through an electrode that you can apply to, let's say, neurons locally. You have no control over which neurons are being activated, and still in deep brain stimulation, when you do that in the human brain, you don't know exactly what you're doing. Now with optogenetics, you have the genetic identification, the selectivity that you can use to activate particular neurons and not others. And now it becomes powerful to dissect synaptic circuits. You can see, let's say, for example, this higher order thalamus. If I activate that, like at will, with light, what happens with the neurons in the cortex? Are they all starting to fire, or they are being silenced? So we can now get these one-to-one relationships, and then go into the synaptic components of it, and start to study, what happens at the synaptic level. So yes, incredibly powerful, and I wish sometimes that people would do more of that rather than really going into the probing of behaviors, because this is also very, very interesting, but there's also still so much interesting stuff to do at the circuit level, I think, with those tools.
And it's also very complex to draw your conclusions about the behavior, right? There are so many circuits activated, play a role. It's a tough one, but important, as you mentioned.
Of course, but it's cute, right? When you shine a light on and you inhibit particular neurons from being activated, and suddenly the mouse has forgotten something, or suddenly it fails to execute a behavior that it normally would do perfectly fine, and thereby showing that this area or these neurons are important in that stream of or in output relationship, but. I mean, in a way, then it is still a bit of a black box, right?
There's still things going on in the background.
But okay, that's fine. It's fine. I'm definitely not dismissing it.
It's actually very interesting. Can you elaborate a little bit more on the technique of optogenetics, since we talk about it?
Yeah, well, I'm not a specialist on it, but it's an interesting optical trick, right? You have proteins. Let's keep it simple. But you have proteins that are gates for ions, but they're sensitive to light. So as similar to what happens in your retina, for example. Although those are not ion channels, but in a similar way, the channel opens when it's activated by light, and then ions flux, and you have an ion flux, and if this ion flux is big enough, you will create action potentials in a cell, and or you can vice versa. You can conversely prevent it from happening. So now it becomes then interesting to come from outside of the brain with light, you can control the activity of the neurons remotely and non-invasively. So it's very, very powerful new development that, again, comes out of almost serendipitously from different fields, right? That it's the cross-hybridization between fields that, again, goes to show how important fundamental basic research is, and we need to keep stimulating that, because this is where these type of developments come from. It hasn't been done by a neuroscientist who thought, okay, let me develop or engineer a probe that can do this. It was bottom up. It was because those channels were discovered and then engineered, and then it turns out to be useful for neurons. A bit unexpectedly at first.
It's fundamental research is indeed very important. I totally agree on that point, and especially in neuroscience, that is a field it's super complex with so many multidisciplinary aspects. You have the computation, biology, molecular biology, biochemistry, even physics is involved, and math, and it's incredible. And totally agree, we need to push even more fundamental research and make aware of people out there that this is actually important. It's not a waste amount of time and money, right?
And even these chemogenetics tools and optogenetic tools that were actually really purposely engineered from a neuroscientific with a neuroscientific goal or from a neuroscientific perspective, rely on very basic fundamental knowledge about like what ion channels do, what G protein coupled receptors do, how they operate, and this is all very basic stuff that then comes to use.
So we talked about tools to study memory in animals, and mostly we talked about processes that are in the healthy animals. However, we have disease in our brains, right? And some aspects are very interesting when you relate them to memory. For example, hallucinations in schizophrenia or hypersensitivity in some autistic patients. What is the underlying? Do we know the underlying principles of this kind of disease in terms of, let's say, the memory aspect?
Unfortunately not. But I would also be very careful in typing it as disorders or diseases. Yes, it's it's different from let's say the average population, perhaps. But some elements of these traits are in all of us, and I think and we sometimes can even recognize ourselves in certain behaviors that some individuals show in the more extreme version, right, or in extreme ways. So I really like to see it as a continuum, and I think I find it important also to look at it from that angle when we are studying it. I think too often we are hunting for some sort of aberration, in a circuit. And it might just be very subtle. It might also change from day to day a little bit. Certainly there are going to be, in more severe forms, there are going to be some very basic changes that are not easily overcomable. But the symptoms that you just described, like, for example, a hallucination, you can create in many different ways. You can temporarily create them. It's not only, an attribute to, let's say, schizophrenic. So, and that is, I find interesting, because I think we should probably look at elements, right, of our behaviors and try to get to understand those. Try to model these kind of aspects in animal models rather than the entire disease holistically, because there is no schizophrenic mouse or autistic mouse for that. That would again be too much anthropomorphizing and trying to see all these different traits into a mouse model, which probably cannot just exist because it's simply not a human being. And then coming back to that continuum, I think when you see it as a continuum, you can also approach it better, I think, in trying to understand what the circuit mechanisms for certain phenomena are, or the lack of particular phenomena. I really am convinced that we're not going to get there otherwise. I think we have done plenty of genetics. There's nothing, right, that comes out, that jumps out, and there's always some slight correlations, and maybe that can be termed risk factors. But no, if there was something that clear, we would already found it with the amount of money and time and effort that has gone into it. So I think we really need to go back to the basics of the elements of the elementary aspects of behaviors and try to understand that, and then maybe build up our knowledge from there and see how that would fit into particular, conditions that we would call.
I think this is a very, very valuable opinion, and if I may add something. So we need this to go back and to see really the fundamental principles, but also I think that at some point we need to change our the way how we look at things, right? There should be some, especially for example in neurodegeneration, there you have this, let's take Alzheimer, you have this two kind of concepts, the tau, a beta that are going on, and people were like running in circles, I think, and it seems that none of the others is that crucial. There is some role, of course, that this kind of protein that are not functioning properly. Let's say they contribute to the disease, but there is something underneath that we still do not understand, and we probably as young scientists need to think about also novel approaches, right?
Absolutely. I think with neurodegenerative diseases, of course, we have a much clearer picture of what is going wrong, right? We see neurons losing functionality and even dying, and so we cannot deny that's not important. Now, where it all starts and how that is related to our to how we live and how we behave, that is still mysterious. And again, I think you're right. We probably are, once we have formulated hypothesis, we like to get that confirmed, and we are hunting a little bit always in the same hunting grounds and trying to chase things that maybe at some point we should accept that it's not worth chasing. But with these philosophical discussions, you quickly step on toes, and there is no one way to do it. And I think, that also in a way that's the power of a scientific community as we know it. We have, there's room for creativity, and someone does this, another person does that, and at some point, cumulatively, like the communal intelligence that we then produce will provide answers at some point. As long as we're not trying to convince each other all the time that, this is the only way, because then I think we're starting to get a little bit too much, all of the same type of research. That said, but maybe that's more like another topic, but we also need to collaborate perhaps more than we do, because the approaches are getting more and more complicated. The questions probably as well, but even simple questions require very, very complicated, sophisticated techniques, especially in neuroscience nowadays. And I think, I don't see it's not feasible. I think that we keep doing it at very small scales. Just simply maintaining all your lasers and all your equipment as a small group. It's difficult.
It's indeed, I totally agree to this perspective, and it's not possible. Not only that it's meaningful and necessary, but it's just not possible anymore to be isolated and be successful. It's just the tools are super complex, the instruments are expensive, the analysis is very time consuming, and completely different topic.
This is because you mentioned tools. I mean, here we have a tool that recently has taken off. Like our ways of data analysis are just, I mean, it's a small lab. As a small lab, it's very difficult to really have that breadth, and that bandwidth to actually do all of that, let alone just to understand all of it. The machine learning, AI that is part of it now. So we definitely need to collaborate, and hooray for more interdisciplinary type of research.
Professor Holtmaat, I would like to talk to you more about synapses and brains and memory, but we are approaching the end of our discussion, and I would not torture you anymore. It's quite late already, and you traveled today. So I would like to thank you very much once again for the time and the great discussion that we had. And I hope that probably in the future we meet each other somewhere at some conference or we collaborate.
Hopefully,
we don't know.
Thank you very much. It was a pleasure to be here at this table. I think I blended quite well in the blue environment here. So, well, thank you. Thanks again. I'm also looking forward to further interacting at this symposium.